Okay, here we are again. This is chapter 10, and in this lecture, I'm going to follow up from some of the information that I presented in the chapter 9 lecture. If you remember, in chapter 9, I described a variety of genetic manipulations, including transgenic mice, where a gene could be overexpressed or knocked out or modified in some way to make it more or less effective or to even make it do something different than it originally did. I also described that we even have inducible and tissue-specific transgenes. In other words, we can cause our gene manipulation to kick in only at a time of our choosing, or we can engineer gene manipulations that will only be expressed in particular places, for example, in one specific region of the brain. So, what I'm going to do today is to describe some really important experiments that have utilized these procedures to manipulate genes, sometimes only in particular circuits in the brain, so we can learn more about the roles of specific circuits in the behavioral responses that we're interested in. So I suppose I actually have two goals overall in this lecture. One is to illustrate some examples of the ways in which genetic manipulations have been used to explore circuits in the brain, and the other is to advance our understanding of the early stages of memory formation in relation to the dynamics of NMDA receptors and AMPA receptors. And a large part of the lecture will focus on one of the most famous experiments ever in the neurobiology of learning and memory, perhaps actually made famous, at least in part, by the creative name the researchers bestowed upon their genetically manipulated animals. So we'll get to that in a few minutes. Meanwhile, let's talk about the role of NMDA receptors in hippocampal circuits. But first, a little background based on a study performed by Sun, Shipley, and Lido in the year 2000. We already know that the NMDA receptor is a heterotetramer made up of a homodimer of GluN1 subunits and either a homodimer or a heterodimer of other subunits. And I think I previously told you that the GluN1 homodimer is obligatory. So I think I told you, but just in case I didn't, I'm going to tell you again. The GluN1 dimer is obligatory. You cannot construct an NMDA receptor without one GluN1 homodimer. But the other subunits vary. There's a choice between having GluN2A or GluN2B or GluN2C or GluN2D or GluN3A or GluN3B. These subunits can form homo or heterodimers, and any one of these dimers can complex with a GluN1 homodimer. However, what Sun and colleagues identified is that the choice is not random, but the different receptor isoforms are regionally distributed. They used quantitative reverse transcriptase polymerase chain reaction. And yeah, I know that's a mouthful, but in other words, RT-PCR, to examine the expression of messenger RNA for NMDA receptor subunits in homogeneous of brain tissue dissected out from a variety of cortical regions and olfactory bulb of young rats. Unfortunately, they were still using the old nomenclature for the subunits in their published papers, and so that's what shows up on the slide. But you will remember that NR1 equals GluN1, and NR2B is GluN2B, and so on. So you can see on the graphs at the left that the GluN2B subunit, for example, is abundantly expressed in entorhinal cortex that overlies the hippocampus, as well as visual cortex, and olfactory bulb. And I skipped over also motor cortex, but not so much in piriform cortex. So you see some regional differences. And it turns out actually that the GLUN2C, 2D, and 3 subunits are not expressed very much in the forebrain at all. Subsequent studies, and you can see those numbers here in these various cortical regions and olfactory bulb. But subsequent studies have shown that these isoforms, the GLUN2C, 2D, and 3, they're found in greater abundance in the hindbrain, including the cerebellum. And so just to orient everybody to where these structures are in the brain, I labeled them in the diagram at the lower right of the slide. And it also has the hippocampus ghosted in, with the entorhinal cortex lying just over it. And so you can see the anatomical localization of these cortical areas in this diagram. So with that background, let's look at the manner in which Zhou Qian and collaborators used a specific targeting strategy to explore the role of NMDA receptors in LTP and spatial learning. Amazing as this may sound, using recombinant DNA technology, they engineered transgenic mice that would have the GLUN1 subunit of the NMDA receptor knocked out specifically in the CA1 pyramidal cells of the hippocampus. And we know again that the GLUN1 is obligatory. So the transgenic animals would have a total lack of functional NMDA receptors in the CA1 pyramidal cells. Now, the sections through the hippocampus at the left show that the cells are healthy, and the overall morphology of the hippocampus actually looks fine in all 4 lines of mice. In these close-ups, the cells are all counterstained with cressyl violet, so you can visualize the cells based on that violet signal. However, the researchers also engineered their lines of mice so the cells would exhibit positive XGAL staining in all cells that express the GLUN1 subunit of the NMDA receptor. This is a dark blue stain. The top figure shows no XGAL staining in CA1 pyramidal cells of the mice in which GLUN1 subunits were specifically knocked out in this region of the hippocampus. All you can see there is the violet stain from the cressyl violet. The other 3 control lines of mice have abundant expression of GLUN1 subunits in their CA1 pyramidal cells. Now, Qian and colleagues then attempted to induce LTP by applying tetanic stimulation to the Schaefer collateral inputs to the CA1 pyramidal cells. The top graph shows that LTP was induced very nicely in the 3 control lines, but not in the CA1 GLUN1 knockouts. And the bottom graph actually shows that LTP was successfully induced by tetanic stimulation of perforant path inputs to the dentate gyrus granule cells in all 4 mouse lines, as you might expect, as the knockout was shown to impact the CA1 cells only. So the CA1 specific NMDA receptor knockout mice were impaired in LTP. So you might wonder if they were also impaired in spatial learning. And well, good for you for wondering that. Of course, Zhou Qian tested this possibility in the standard Morris water maze. They found that the CA1 specific GLUN1 knockout mice improved across training trials, shown in the closed circles here, but were actually slower than the other 3 strains of mice to actually find the hidden platform. But they were okay pretty much on the visible platform version of the maze. In other words, the knockout mice were impaired in the spatial version of the test, but not in the non-spatial version. But most importantly, the probe trials revealed a substantial deficiency in the knockout mice, as they failed to explore the target. You can see the 3 strains of control transgenic mice all preferentially explored the target quadrant, but the CA1 GLUN1 knockouts, represented by the black bars here, explored, well, all quadrants equally. So this shows us that even if part of the hippocampal circuit is intact, we saw the perforant path to granule cell component was intact. And hypothetically, the mossy fiber to CA3 component should be fine too. But even if those parts of the hippocampal circuit are intact, specifically knocking out NMDA receptors in the Schaefer collaterals to CA1 pyramidal cell synapses is enough to disrupt spatial learning in the Morris water maze. Okay, so let's look at an interesting contrast. Rather than knocking out the GLUN1 subunits in the CA1 pyramidal cells, as Qian and colleagues did, Nakazawa and colleagues selectively knocked out GLUN1 subunits in the CA3 pyramidal cells. Now, Nakazawa and colleagues didn't investigate the impacts of this manipulation on LTP, but they did test in the Morris water maze. And they found that the CA3 GLUN1 knockout mice did just fine in the standard spatial version of the test, where the location of the platform was held constant across learning trials. Unfortunately, they didn't report probe trials to examine the memory of the mice for the location of the platform. They just reported these learning trials. However, when they increased the difficulty of the task, using a delayed matching to place version of the Morris water maze, the CA3 GLUN1 knockout mice didn't do so well. In this version of the test, the location of the platform was moved each day, and the ability of the rat to swim to the novel place of the platform was assessed across repeated trials each day. And you can see right here that these CA3 GLUN1 knockout mice did not find the platform as quickly on each of these days as the wild-type mice did. Accordingly, it seems that disruption of the mossy fiber to CA3 potentiation wasn't quite as debilitating as disruption of the Schaefer collateral to CA1 connection in this circuit. But still, disruption of this component of the circuit has detrimental consequences, interfering with acquisition of more complex spatial calculations. And this suggests to me that different links in this circuit actually have different functions. We're not just relaying information from one synapse to another synapse to another synapse. We know that there are recurrent processes within the hippocampus. There's communication between the 2 hippocampi and back and forth with other parts of the forebrain. And so each of these connections within the hippocampus may be playing slightly different roles in the incorporation of spatial learning and memory. So different synapses in the hippocampal circuit that we know is so important for spatial learning may be playing different roles in the actual encoding of that learning. But the next thing I want to do is try to build a case that different subunits of the NMDA receptor may also provide different contributions to memory formation. We know that different parts of the brains of mammals express different combinations of subunits of the NMDA receptor. Again, the GLUN1 is obligatory, but the GLUN2 subtypes can vary from one part of the brain to another. So here are two interesting points. One is that this differential distribution of GLUN subunits is not only true in mammals, but it's also true in other vertebrates, like birds. Now, from an evolutionary perspective, this suggests that the differences in expression of GLUN2 subunits in different parts of the brain plays a very important role in species survival. Otherwise, why would we see this phenomenon in all the different groups of vertebrates that have been studied? It must be important. And the other interesting point is that across species, including birds, there's actually a developmental shift in the expression of specific subunits of the NMDA receptor in the forebrain. In particular, the GLUN2B subunit is expressed highly in forebrain regions in juvenile development in mammals. Later, there's a switch to GLUN2A subunits. And this is illustrated in the diagram at the upper right of the slide. This schematic shows early in development, we've got not so many of the GLUN1, GLUN2A heterotetramers. But later in development, there's a switch so that we've got more of these GLUN1, GLUN2A subunits squeezing out those GLUN1, GLUN2Bs that were expressed so much in earlier development. You see that shift from lots of GLUN2Bs early on to not so many GLUN2Bs later. But there's also an interesting version of this switch in canaries. Okay, so most songbirds learn their species-specific song in the first year of life, and then sing it when they're mature, the first mating season at 1 year of age. But canaries are a little different. They are actually seasonal learners, and they relearn their song every year. If the learning is disrupted in any year, the song doesn't crystallize. Amazingly, they actually revert to the juvenile form of NR2B receptor expression every winter when they're relearning their song. And this is especially true in the song encoding areas of their brains. Perhaps even more amazing is that the expression of the GLUN2B to GLUN2A ratio can be altered simply by changing the duration of light in the daily light cycle for laboratory-housed birds. When you shorten the light period each day, imitating winter, you get higher expression of GLUN2B subunits in the song-related brain areas. Isn't that cool? Okay, but based upon this and other bits of information that were available about the GLUN2B subunits and the switchover to GLUN2A later on, Yaping Tang and Zhou Qian's lab examined the possibility that the GLUN2B subunit actually conferred a learning advantage. Now, there's one more thing I haven't told you about the difference between the GLUN1, GLUN2A and the GluN1, GluN2B NMDA receptors. Electrophysiological experiments using patch clamp technology, which can characterize the actual current running through an individual ion channel and even isolate what kind of ion is exchanged, has revealed that the GluN1, GluN2A forms let in a reasonable amount of calcium each time glutamate binds when the postsynaptic neuron is depolarized. And I had that depicted really nicely on the slide here. I'm going to draw it back in for you. Here's your GluN1, GluN2A receptor. VoiceThread keeps doing this to me where it distorts any kind of drawing that I've tried to put in there. So I've caught a few of them, but this one snuck through. This should be another unit in this side here, making the GluN2A component of this NMDA receptor. And I actually originally had a really nice animation going here. So it showed that when glutamate binds and the postsynaptic cell is depolarized, that all of this calcium comes running in. It actually showed it kind of coming through here. But we've got a reasonable amount of calcium coming through this GluN1, GluN2A receptor. Okay, but on the other side over here, we see that the GluN1, GluN2B stays open for a longer duration. When glutamate binds and the postsynaptic receptor is depolarized, this NMDA receptor stays open for a longer time, increasing both the window of coincidence detection and allowing a much greater influx of calcium. And again, the original animation kind of had this GluN1 and GluN2B opening up to open the ion channel, and then I got a whole bunch of calcium running through here. So I had to modify that a little bit since VoiceThread doesn't do those kinds of animations, but you get the picture. On this side, the GluN1, GluN2A lets in a little bit of calcium and closes up quite quickly. On the other side, the GluN1, GluN2B, under these conditions with depolarization and glutamate binding, opens up wide and lets lots of calcium through. So of course, Zhou Qian's group suspected that this may help to explain the phenomenal learning abilities of human children, and canaries, I guess, and other juvenile or seasonal learning animals. So they set out to test this hypothesis using genetic manipulations in mice. So GluN2B subunits are abundantly expressed in youth, and GluN2A subunits abundantly replace them in adulthood. Now, Qian and his group reasoned that if they could overexpress the GluN2B in adulthood beyond the normal developmental window, they could create a super smart mouse. And, well, indeed, that's what they did. Remember that I said I would talk about a very famous experiment that was at least partially famous because the researchers gave the mice a really interesting name? Well, this is it. Tang and Qian called their super smart mice Doogie mice, named after a very bad early 1990s sitcom called Doogie Howser, MD. Get a load of this. It shows just how bad TV was back then. Doogie Howser, MD was the story of a 12-year-old super genius who graduated medical school and was now practicing medicine at the local hospital. Yeah, like if I went to the hospital and a 12-year-old boy was the doctor assigned to my case, I'd be thinking, how lucky am I to have such a smart doctor? Not. Anyway, it did launch the career of Neil Patrick Harris, shown on the left over here as 12-year-old super genius Doogie Howser, and more currently in the photograph over on the right here, as you may know him today. But That show not only launched the career of Neil Patrick Harris, I guess it also launched the media sensation of Doogie mice, which were nearly as popular as their namesake about 6 or 7 years later. Anyway, the Doogie mice were genetically engineered by adding a gene construct to laboratory mice. The gene construct consisted of a CAM kinase 2 promoter with an extra copy of the GluN2B encoding region downstream from it. This takes advantage of the fact that CAM kinase 2 is abundantly expressed in neurons of the forebrain throughout life, or at least starting shortly after birth and then throughout life. And it takes advantage of the fact that the GluN1 proteins that are encoded by a different gene assemble in the membrane with whatever NMDA subunit they happen to bump into. If there are lots of, let's say, GluN2A proteins, they will assemble into GluN1, GluN2A type NMDA receptors, which is typically what happens in adulthood. But if there's an overexpression of GluN2B subunits, then those GluN1 proteins will more likely encounter them in the vesicle membrane and will more likely assemble into GluN1, GluN2B receptors. So that's the construct that they put together. And after they created these mice, they screened them for any abnormalities, which is very important when creating new transgenic mice. They found that they had normal physical appearance and body weight, and they had normal behaviors in relation to anxiety and other measures. And they didn't exhibit seizures or any other indicators of CNS abnormality. They also looked at gross and fine markers of the development of the brain. They found that they had normal brain structures, including the morphology of the hippocampus shown over here in the upper right, and they had normal cells, including normal dendritic spine profiles in comparison with those of wild-type mice, shown in the center of the slide. But using in situ hybridization, they could tell that expression of the GluN2B subunits was upregulated in the forebrain, perhaps especially in the hippocampus, cortex, and amygdala, shown in this figure at the bottom right. In the Western blot analysis shown on the upper left, we can see that the protein expression for GluN2B is upregulated in the hippocampus and cortex for the 2 strains of transgenic mice in comparison with the amount of NR2B seen in the wild-type mice. The expression of GluN2A subunits also seem to be upregulated in the hippocampus and cortex of the transgenic mice in comparison with the wild-type mice. But the expression of the GluN2A subunits was unchanged, or no differences between the 2 strains of Doogie transgenic mice and the wild-type mice. So this suggests that overall, we've got an upregulation of GluN2B subunits in the transgenics, and we've got an upregulation of the GluN1 subunits, which suggests that we perhaps even have an overall increase in the number of NMDA receptors expressed in cortex and hippocampus of these animals. And at the lower left, northern blots that show mRNA expression was upregulated in tissue homogeneous that included a whole bunch of tissue from cortex, striatum, and amygdala here, And from hippocampus here,